Hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. You're joining the three NIH-funded National Cryo-Electron Microscopy Centers. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. And here with me today from NCCAT are Ed Eng and Mahira Aragon. Joining from the Pacific Northwest Cryo-EM Center, PNCC, is Sean Mulligan. And from the Stanford SLAC Cryo-EM Center, S2C2, is Michael Schmid. Today's speaker is Keerthi Gottipati, joining us from the University of Indiana, Bloomington. And I'm going to let Mahira introduce her properly after a quick update from each of the three centers to keep you up to date on the resources we make available at no cost to the research community. This webinar is a regular event. We host the last Thursday of each month to highlight the methods and tips and tricks that groups are using to collect and interpret the data they can obtain with the help of National Cryo-EM Center resources. You can save the date already for our next talk in July. Virender Singh from UT Southwestern Medical Center will present on structural polymorphism and ATTR amyloidosis. So if you're interested in that, the registration link is already available on the events page at cryoemcenters.org. And you'll also find recordings of all of our past talks there as well. One more announcement from me before we get to today's business. Summer is conference season, and staff from the centers will be at the ACA Structural Science Society meeting in Denver, coming up in just a week and a half, I think. And we'll also be at the Microscopy and Microanalysis meeting in Cleveland. So if you'll be at either of those meetings, come say hi. If you don't find us wandering the halls of the meeting, we do have booths in the exhibition hall, so it will be easy to find us. Back to today, please send questions at any time using the Q&A box. That's where we'll be watching for them, and we'll moderate questions at the end of the talk. So let's go ahead and get started. So first, I will turn the mic over to Sean to tell us what's new at PNCC.
Thank you, Christina. Good morning and good afternoon, everybody. I'm Sean. I'm a microscopist at the Pacific Northwest Cryo-EM Center. PNCC has two types of proposals: screening access and general access. Screening access allows you to come on site for up to 5 days of sample preparation and screening with staff assistance. This proposal is aimed at low resource institutions. Our other proposal type, general access, awards up to 480 hours of microscope time a year for 2 years. This time can be used for screening or data collection. Approved general access proposals are also eligible for our training workshops, which cover topics from sample preparation through data processing. Approved proposals are also assigned a scientific point of contact, or SPOC, such as myself, who can help you with questions about sample optimization, data collection strategies, and so on. Our center has an Arctica, a newly installed Glacios 2, and 4 Cryos. 3 out of our 4 Cryos have K3 direct electron detectors with biocontinuum HD energy filters. One of our Cryos was recently upgraded to a Falcon 4i with Selectress energy filter. And for sample preparation, we have a few Vitrobots, Leica EMGP2, Vitrojet, and manual plungers. If you're interested in applying, our deadline is the 1st of every month, and we look forward to hearing from you.
Thank you, Sean. Now I will turn over to Mike to tell us what's new at S2C2.
Hi, I'm Mike Schmid. I'm one of the co-directors of Stanford SLAC Cryo-EM Center and of the Stanford SLAC Cryo-ET Specimen Preparation Centers. We have a very busy summer where we are taking the acceptance test on our 4th and final Cryos, brand new Cryos, which should be available in August and September to support our other 3 Cryoses. We also have, we're also installing, as we speak, a Glacios and a Tundra to support our screening and sample optimization missions. We just did our acceptance test of the first bio Hydra fib milling instrument that supports the specimen preparation center for Cryo-ET. So if you're interested in either single particle or Cryo-ET, look us up.
Thank you, Mike. And I will turn over to Ed.
Hi, I'm Ed, coming here from New York City, where NCCAT is located. We have been doing a lot of capacity building like the other centers. Pretty soon we'll have 5 Krios instruments, 2 Glacios instruments, a Tundra dedicated for instrumentation access. We offer high-end data collection, screening, and cross-training. In terms of the summer conference season, if you are an ACA or M&M, we're going to be hosting a meetup. Both of them are going to be lunches. If you want to sign up, either email Christina or myself or indicate that in the chat. If you want to find out more, go to nccat.nysbc.org.
Thank you, Ed. I will stop sharing. Mahira Aragon is a vital member of the team at NCCAT. She works with most of the users who come here to use our chameleon blot-free vitrification device. And she worked with Keerthi when she came here on a chameleon proposal. And I'm going to let her introduce today's speaker.
Okay. Hello, I'm happy to introduce today's speaker, Dr. Keerthi Gottipati, whom I had the great opportunity of working with. She is currently an associate scientist at Indiana University in Bloomington. She has used structural and biochemical studies to understand viral proteins participating in replication and immune evasion of positive strand RNA viruses, starting with her graduate training at the University of Texas Medical Branch in Galveston. During her time in Texas, she received training at the Cryo-EM, cryo-electron microscopy laboratory in Cryo-EM sample preparation, handling, and imaging. She uses Cryo-EM and X-ray crystallography in conjunction with RNA engineering to study viral RNA, protein, and RNA protein complex structures. With this experience, she contributed the first reported high-resolution structure of the enterovirus cloverleaf RNA, recognized by a U.S. National Science Foundation Conference Award to support training and innovation in RNA science at the 2021 RNA Society meeting. Her work is also supported by seed grants from the University of Texas Medical Branch and Indiana University. Our long-term goal is to develop a template for rational drug design based on macromolecular structures of RNA and RNA protein complexes that participate in viral disease pathogenesis. At the time of her NCCAT visit, her project focused on the viral RNA protein complex, HIV-1 Rev response element, RNA, and HIV-1 Rev protein. Today, we are excited to hear about the progress of this project. Thank you for your time and allowing us to learn about your project.
Awesome. Thanks, Myra. So, okay, I'm just gonna, this is the progress report of what we've done so far with the HIV Rev response element. So before I dive, 1 second, this is not moving yet. Oops. Okay, before I dive into the actual research subject, I just wanted to talk a little bit about viral RNAs and their role as regulatory elements. Viral RNAs Yeah, so viral RNAs are not just passive coding sequences for proteins, but they also act as regulatory elements that regulate the viral replication, translation. So I'm just listing a few of their functions. One of them being they could be promoters in positive strand RNA virus replication, like the 5' and 3' UTRs. Let's start over. I'm so sorry. Okay. These can, here are some of the functions of the viral RNAs. They could be promoters in positive strand RNA virus replication. These are structures such as the 5' and 3' UTRs in flaviviruses. They could be the internal ribosomal binding site, internal ribosomal entry sites, which are ribosomal binding sites, and they promote viral translation. They can be transcription and translation signaling. involved in transcription and translation signaling. This is done by the cloverleaf RNA in picornaviruses or the frame-shifting pseudoknot in SARS-CoV-2. Also, another example is the viral packaging signal, which is seen in HIV or in the HCV cis-acting duplication element. So I guess the main idea here is that RNA is so flexible and able to interact with other proteins and other RNAs and able to fold into secondary and tertiary structures that it can be involved in a bunch of these functions. And viral genomes, owing to their small size, use this nature of RNA to pack a lot of function into it. So it's not just a coding sequence, but a regulatory element as well. So this brings me to the RNA that we work with, which is the Rev response element of HIV-1. This is involved in the translocation of the RNA from nucleus into the cytoplasm. So before I talk about the exact function of Rev or RRE, I wanted to give you a brief overview of the life cycle of HIV and where this RNA comes into picture. So HIV is a lentivirus. It belongs to the Retroviridae family. It's a positive strand RNA virus and contains 2 copies of its genome packaged into the virus particle along with the reverse transcriptase protein. So the life cycle of HIV can be divided into 2 halves. One involves the establishment of persistent infection, which ends in the genome being integrated into the host DNA. The other half is when the virus is actively transcribed, replicated, and packaged into the progeny virus. So the virus initially recognizes the receptors on the cells. It is endocytosed. The membrane of HIV fuses with the cellular membrane. The genome is delivered into the cytoplasm as a nucleocapsid core. Once the genome is delivered, the RNA, the positive strand RNA is reverse transcribed, and you have the formation of the double-stranded DNA, which is translocated into the nucleus. The double-stranded DNA is then integrated into the host genome. That's when you have the establishment of persistent infection. This proviral DNA, after integration, has to be actively transcribed, and the viral transcripts need to be moved again into the cytoplasm where they are translated, and then you have viral assembly and packaging, and you have the budding of the new mature virions. So, okay, so a little bit more about, I'm sorry, this slides are not, there you go, okay. All right, so there's a little bit about the HIV genome itself. It's a 9 kb RNA genome, and yet it produces 15 viral proteins. The way this HIV is able to do this is through different open reading frames and also alternative splicing. So you have when the RNA inside the nucleus is, or the DNA inside the nucleus is transcribed, you have viral RNA transcripts that are fully spliced, which give rise to these 2 kb mRNAs. And then you have the partially spliced RNA, which is about 4.5 kb transcripts. And then you have the unspliced RNA, which is the full-length genomic transcript. So in the early stages of viral replication, or sorry, when these RNAs are transcribed, the fully transcribed viral RNA exits the nucleus. Because it does not contain any intronic sequences, it's freely able to exit the nuclear pore, and you have the translation of the regulatory proteins, Rev, TAT, and NEF. The other 2 transcripts, which are 4 and 9 kb long, still contain the intronic sequences, and these are sequestered inside the nucleus. Later on, after you have sufficient accumulation of Rev in the cytoplasm, Rev translocates into the nucleus, binds the intronic sequences within the unspliced and partially spliced RNAs, and it's able to translocate these RNA transcripts into the cytoplasm. At that point, the RNA is translated, and you have the expression of viral assembly proteins, the GAG protein, and then viral polymerase. So this interaction of Rev with the intronic sequences allows for the partially spliced and unspliced RNA to be translocated into the cytoplasm. So this is a critical step in the establishment of this replication cycle. The way this happens is when the HIV-1 Rev binds the intronic RNA, in our case, this is the RNA of interest, which is the Rev response element, and it facilitates this whole translocation of these RNAs into the cytoplasm. So that brings me to the Rev response element itself. So what is RRE? So RRE is a 350 nucleotide intronic sequence, which is highly structured. Depending on which sequence and which sequence we look at, it can be 4 or 5 stem loop, and even within the same sequence, it can exist as 4 or 5 stem loop structure. It's present, the coding sequence region of RRE is present within the ENV, the envelope protein coding sequence. Like as I said before, it's intronic. And when you have in certain open reading frames, or rather in the fully spliced RNA, RRE does not exist. As for binding of Rev to RRE, the stem loop 2 contains the initial high affinity binding site for Rev. Rev is also able to bind other stem loops in RRE, but 3 through 5 are not essential for Rev interaction. And stem 1, which contains the other high affinity site for Rev, it has been suggested that it stabilizes the Rev multimers. So basically, I'm showing you here the proposed secondary structure of RRE. You have stem 1, the stem 2, and 3, 4, and 5. Okay, and then Rev itself is a 116 amino acid long protein. It has an N-terminal domain and a C-terminal domain. The N-terminal domain is highly structured at 70 amino acids. It contains a nuclear localization signal and the arginine-rich motif. The arginine-rich motif is responsible for the RNA interaction. It also has oligomerization domains. These are involved in both dimer and oligomer formation. And then the C-terminal domain contains the nuclear export sequence, but it's mostly unstructured. This is the NMR structure. The structure of Rev. Rev has been, the structure of Rev has been, Rev 70 has been solved a few times, and the dimer can exist in 2 different conformations. This is one of those. As again, the color coding is according to what's shown here. So you have the oligomerization domains, and this helix loop helix region would form the arginine-rich motif and the RNA interaction region. So, so what do we know about, at least structurally, what do we know about the HIV, RRE, and Rev interaction? So far, we do not know the full length structure of the Rev response element. We have some information about the high affinity binding site. There was the structure determined of Rev in complex with the stem 2B of RRE. Stem 2B contains the high affinity site, and this was a minimized section of stem 2B, not the entire stem 2. And we see Rev binding within the major groove of this stem 2B structure. We still do not know how Rev interacts with the rest of the RRE. we do not know how Rev binds and oligomerizes onto the RRE sequence itself. We know that the structure of RRE is required, or rather it directs the oligomerization of Rev onto oligomerization of Rev, and then its interaction with the nuclear export complex. So this is just a cartoon representation of dimers of Rev bound to the rest of the Rev response element. So right now, because of a lack of any structural evidence, even though there's a lot of research already done on this complex, we still do not know the nature of this oligomeric complex. So we set about trying to determine this by working with the RNA first. So the thing about RNA structures is so far only a very small fraction of all the available structures on PDB are RNA-only structures. The properties of RNA that make it so useful and so interesting to work with, that is the dynamism, the flexibility, its interaction with other proteins. They're also the reason why it's so hard to work with RNA, at least in terms of structure biology. So you have either, because it's only 4 bases, you have signal redundancy, or rather signal overlap. And then because it's so flexible, RNA can be hard to crystallize. And it's also can be hard to work with in cryo-EM samples because it's so heterogeneous, and and you have low contrast in the images. So one way we are trying to approach this problem of getting an RNA structure is using the tRNA scaffold approach. This is just an overview of the tRNA scaffold. We replace the anticodon stem of the tRNA with the RNA of interest. And this chimeric RNA is expressed in E. coli as just as you would express any recombinant protein. And then we do just a one-step purification of this. It could be as easy as phenol chloroform extraction, and then you do ion exchange chromatography, and then yes, you have voila, a good clean sample of the RNA. So there are several advantages to this approach. First thing is when you do a T7 transcription, you always end up with extra nucleotides at the 3' end. The tRNA scaffold eliminates that issue. We have a homogeneous 3' end. It prevents the unwinding of the double-stranded RNA helix. So at least we are knocking out the conformational heterogeneity on one side of the RNA sample that we are working with. We produce it in E. coli, and we get large quantities of the RNA. So it's useful for both crystallography and cryo-EM. The cost effectiveness works too. And then especially in crystallization, tRNA facilitates crystal contacts, and you have the probability of crystallization increases because of that. And then in cryo-EM, tRNA serves as like a reference point to orient your maps or models when we build them in. Oh. Okay, and then so using this scaffold approach, we did determine structures of a few viral RNAs. We did the dengue virus and Zika virus stem loop A structures. These are the promoter regions that I talked about in the beginning. We did the Coxsackie virus and poliovirus cloverleaf RNA structure. And then we also determined the stem loop 2 structure of HIV RRE, which I'm going to talk about in not too much detail, but in a little bit. So these are, okay, so the stem loop 2 structure of RRE. So while I'm working with cryo-EM, we are also interested in looking at the RRE piecemeal. So we are looking at the high affinity Rev binding site of RRE, which is the stem loop 2. So we recently determined the structure and published it. I'm not going to go into too much detail, but just to say Okay, so that using the tRNA scaffold approach, we were able to crystallize this. And we found that the stem loop 2 exists in two conformations, or we were able to capture two major conformations of this RNA, a closed conformation and an open conformation. They differ from each other at the three-way junction, which also contains the GGG motif, which is the high affinity binding site for Rev. This structure helped clarify the location of the high affinity binding site. It was thought to be part of the stem 2B, but now we know that it's part of the three-way junction. And the way the GGG motif is stacking and interacting with each other determines whether or not it's the conformation is closed or open, which also changes the width of the major groove where a Rev is supposed to bind. So again, I'm not going to belabor into this because it's already published. All right, so using the same approach, this is where we started attacking the full-length RRE. Even though RRE is 350 nucleotides long, the functional part of RRE, or which is required for RNA translocation, is just 237 nucleotides. So we used that and created a tRNA fusion construct, expressed this in BL21 cells. Again, just a regular quinol chloroform extraction, and then we get a clean prep of the RRE. So this is showing you the rest of the cellular RNA that are extracted, and then the clean RRE prep that comes out at the end. And then we did some functional verification, whether this RNA is completely folded. We did a FACS analysis. It's a monomeric species. The Dmax and that was determined using FACS falls along the same line as when we would calculate using just the secondary structure. And then we tested the Rev binding. We looked at binding to just the arginine-rich motif peptide, and then Rev-70. As was seen previously, several copies of ARM and several copies of Rev bind the full-length RRE. So now we know the RNA is clean and it's functional, and so we went ahead and did a Cryo-EM grid preparation. So this is where we ran into all the issues. Because this is a method stock, I'm kind of showing you all the bad pictures as well. So we initially worked with RNA that was suspended in 50 millimolar heaps. We kept the RNA concentration pretty low, and we were doing manual blotting with Vitrobot at 20 degrees and 100% humidity. So I think this is a common problem with most RNA samples, where the RNA it does not stick to the ice, but is targeted into the carbon support around the ice. This is just to show you that we were not seeing any sample inside, and most of it was sticking to the carbon. So to overcome this problem, we tried a few techniques. We pre-treated the grids with polylysine. That's basically because we're trying to neutralize the charge. Maybe since RNA is heavily negatively charged, we thought maybe there's some kind of repulsion. We could alleviate that problem with using polylysine treatment. We treated the grids with detergents. We tried beta OG between 20. The other treatment was with BSA, just coat the grids with BSA and then add RNA to it. We also tried carbon and graphene coated grids. So all of these methods work sometimes. We never saw consistent results with any of these. So this was, and then we would also get RNA sort of aggregation and things like the artifacts. What did start working for us was the multi-blot approach, which is saturating the sample on the grid with a lot of RNA. So VitroBot allows you to blot multiple times onto the grid before you plunge freeze it. So this actually does help with saturating the sample on the carbon, and then eventually it does stick to the ice in the holes. The other approach that also kind of worked, was the protein chaperone approach. So for this protein chaperone, we used the Fab antibody fragment. So this was something that was developed by the Piccirilli group. They developed Fab BL36 as a antibody fragment that binds RNA. It was designed as a protein chaperone for RNA crystallography. It binds to the AAA-CA RNA pentaloop, and then with about 100 nanomolar affinity. So I mutated this loop region on the stem 2C to the Fab binding site. And then we tried to form a complex of RRE with Fab, which actually forms a really good complex, and tried to visualize these in using Cryo-EM. We could not keep the complexes together. So the other way this worked for us was pre-blotting the grid with Fab itself. So we would add Fab to the grid, blot the excess protein out, and then add RNA. This is when we saw a decent grids with the RNA being localized to the ice. These two images are trying to show you one is good, one is bad. This is where we have only Fab on the grid. Fab tends to sometimes crystallize on the grid itself, but this is where we have RNA localizing onto wherever Fab was present. So in using this, we were able to get some preliminary 2D classes. We collected data on these grids and were able to get some 2D classes. And then we had like blobby, low resolution reconstructions. We could tell, at least from here, that this was a sample that the conditions were improving, so to speak. And then we could see the tRNA on one end and the RRE portion on the other half. This was also when we started working with NCCAT, because we still don't have a very uniform distribution of the sample on the ice, and we were not able to go any more, and the amount of sample that was sticking to the grid was also pretty low. So I don't have to introduce Chameleon to you, but just to mention that Chameleon is a blot-free approach to vitrification. It uses picoliter sample volumes. It has precise control over the process, and it's completely enclosed. It's fully automated process. The other advantage being that it possibly reduces the air-water interface effects. So this is Maida and me working on the Chameleon at NCCAT. Some of the disadvantages with this Chameleon, as I realized while I was working with it, it requires a relatively high sample concentration. We were using 10 micromolar samples of RNA, and then we can only use it with self-wicking nanowire grids. So that limits where it basically requires special type of grids. And then the area that you wick onto, or where the sample is present, is basically this streak of sample right here, and it's you're limited by where the streak falls, and that's the only usable area. So it's enough for most samples. It may not be enough depending on how good your particles are. And then you have to be very precise when targeting the sample in sections, unlike where you have copper grids where you can choose the edges of the hole. While targeting on nanowire grids, you really want to be at the center of the grid because it just creates some weird artifacts when data processing. So anyway, long story short, we were able to successfully get some good sample freezing conditions, and we saw good sample separation and accumulation on the ice. So with this, we went on to collect data. Oh, okay. So this is, I'm trying to show you that we really do see RNA in the ice. So we collected about 13,000 micrographs, and then so these are the individual RNA particles that we see. So this is going over the Cryo-EM single particle analysis workflow following that data collection. So of the 13,000 movies that were collected, we were left with about 12,000 micrographs. All of this was done in CryoSpark. So one thing that did not give us good results, initially at least, was blob picking in CryoSpark. Wasn't so great. It always worked for me where I created the templates from manual picking, and then template picking would pick up most of the non-junk particles. Even then, after picking and curation and 2D classification, we were left with 1.5 million particles. One thing that was really critical at this point was the several rounds of 2D classification to clean up the samples. I mean, these are common sense things, but I guess at the time it was, for me, a learning curve. Okay, and then so from these 2D classified particles, these are the representative 2D classes. We did an 8-class ab initio reconstruction. We would always get these smaller volumes that we discarded, and then just worked with the long, what looked like full-length volumes of RRE. And then we took these particles and then repeated this approach again, basically, like taking the particles, 2D classifying them, cleaning them up, and then again going back to ab initio and then heterogeneous refinement. So several rounds of this, and then we were left with the 3D refined volume, and we'll end up with about 200,000 particles. So we're really losing about 95% of the whole particle that we pick. So this tells you that you might need at least, it kind of drives home how heterogeneous this sample is, and then how much more data you need to get at a decent reconstruction for this RNA. Okay, and then this is the final model or map that we end up with. It's a 4.8 angstrom map. Kind of shows you how much the distribution of the views we have for this map. It's mostly we definitely seem to have a preferred orientation issue, and then the map looks pretty clean, and then we also see the tRNA end of it and the rest of the RRE end of end. We mostly see that the tRNA scaffold stabilizes the stem 1 of the RRE, and we see a little portion of the rest of the stem loop structure. So again, because it's so heterogeneous and flexible, we think it's averaging out, and we are not able to visualize the rest of the stem loop structures. So in order to fit the model of the RRE into the structure, we also worked with the stem 1 of the RRE in using crystallography. Once again, we made tRNA scaffolded stem 1 construct. We crystallize this construct. So this is the structure of the RRE stem 1 alone. As expected, it has a linear double-stranded RNA structure. However, where this GGG motif is present, which is supposed to be the Rev binding site, we see a kink, and the GGG actually interacts with another G higher up in the structure. So this G46 intercalates with this GGG, essentially kinking the RNA and blocking the Rev binding site. So this intercalation, like I said, blocks the Rev binding site, and we think it presumably acts as a conformational checkpoint for Rev binding. So this structure, we're still writing up this paper, but meantime, we were able to fit this stem 1 RNA structure into the map of RRE. So we have tRNA on one end and then the stem 1. So it fits really nicely with the Cryo-EM map. Even the exaggerated turn happens exactly where this G46 is intercalating with the GGG motif. So in fact, in the full-length RRE, which is the Cryo-EM map, it's even more exaggerated. So essentially saying it's not a crystallographic artifact, it's actually does exist in the RNA itself. This is the model we've come up with so far. We do see a portion of the other stems, but we haven't, that's not clean enough to do any kind of fitting. So in summary, what I've basically shown you is, it's not a surprise, the RNA is very heterogeneous and flexible, but the tRNA scaffold does help stabilize the structure and helps us work with the RNA sample in the Cryo-EM settings. And then the protein chaperone is a valuable approach in overcoming the charge effects and then targeting the RNA to the ice. We might have to fine tune the way we do this with the protein chaperone. The blood-free system is excellent in alleviating sample distribution issues. So while we've been working with, after NC-CAT, we've also been continuously working with this RNA at home to come up with methods to have good sample distribution using normal blotting approaches. So now with a very high RNA concentration, we seem to have overcome most of the sample localization problems. So this is showing you the micrograph that we have, recent data set that we collected, which is at a very high RNA concentration. You can see RNA localized onto the ice, and it's a very clean sample. Yes, it's a little crowded, but we still see very good sample distribution. Okay, so this leads us to basically the future directions. Even though we have alleviated some problems of working with this RNA, we still see a lot of conformational heterogeneity. So we want to work with Rev-bound RRE, which is eventually the goal, but it might also actually stabilize the RNA structure and help us visualize the other stem loops. And the other is to collect data with a stage tilt so that we get different views of the RNA and not just a single head-on view that we've been seeing so far. And that's it. With that, I will end and acknowledge all the universities and people that were involved in this work. So this is in a reverse chronological order. So I started off at UTMB. Misha Sherman was the person who trained me in Cryo-EM. Mark White was responsible for all the crystallography work that we were doing. And then I worked with Myra and Ed and Kashyap at NC-CAT. Now we are at IU. Jericho, the grad student in Kay's lab, was the one who did all the crystallography work. The Cryo-EM Center, David and Giovanni run the center, and they were essentially responsible for getting us started here. And we are finally working with Purdue at this point to collect using their Krios instrument to collect all the data that we screen at the home source. And I will stop and take any questions.
Thank you, Keerthi. That was amazing. And our audience, you can't hear their applause, but I'm sure it's out there. I really have to thank you.
Sorry, I got off to a rocky start, but I hope it made sense.
Absolutely. I really appreciate seeing your initial not perfect micrographs, because that is what everyone experiences and no one ever shows. So that was fantastic. We have a couple questions already. So I'll get those, we'll get started with those, and others can send in questions while you're getting to those. So Miranda had a question about the HIV-1 Rev. And it looks like it has a linker region that connects the oligomerization domain and the NLS sequence. Is that segment disordered or flexible? It looks fairly short and taut in the structure you showed. Are the Rev dimers domain swapped in any of the conformations?
So the Rev oligomerization domain is structured. So the first 70 amino acids are where the oligomerization domain, the RNA binding domain, they all exist, and that part is pretty structured. What is not structured is the C-terminal half. Rev itself is a very small protein. It's only 116 amino acids. So I don't know about the swapping of domains. They're not. It's just a wild type construct with some mutations to prevent oligomerization.
All right, thank you. Another question from Miranda. Are there structures of the, so the structures you showed of the tRNA scaffolded RRE, are they strictly monomeric RRE? Or is there any evidence of dimeric forms?
We are not seeing any dimers. Even when we do native gels, we only see one species that comes out. We don't see dimers.
All right. A question from Varun. Seeing as your particles are elongated, did you encounter overlapping particles at high concentrations, which would effectively lead to noisy density at the terminal? If so, what tricks did you apply in 2D or 3D to solve that problem?
So we do have overlapping particles. We do have particles that are very close to each other. I think I don't know if there are any tricks to actually get rid of this. We pretty much end up losing a lot of particles when we do the averaging and 2D classification, which is probably why we also need a lot of particles to begin with. The 2D classification gets rid of, like this screening at that stage gets rid of most of those particles. Some of the 2D classes, we see two particles in one class, and we get rid of all those classes. And then I guess 3D, what really helped was, instead of 3D classifying, going back to ab initio and then redoing the ab initio models with 2 classes, 3 classes, 4 classes, that eliminated most of the bad volumes, so to speak, while cleaning up the particles.
Thank you. There's a question from Kasahoon. With your positively charged grids, did you incubate your sample for a longer period of time, like 1 minute, 2 minute incubations on the grid?
So when you say positively charged grids, I'm guessing you mean like polylysine coating or things like that. I followed the protocol that was published, and I think it was just you add polylysine, let it sit for 5 minutes, and then you wick the polylysine away. But if we are talking about like multi-blotting or pre-blotting with protein, that I didn't have to wait too long. I pretty much did that within VitroBot itself. So VitroBot, like I said, allows you to multi-blot. You just add the sample, wait for like 10 seconds, and then wick it out and add your RNA or protein of interest. So that can be timed within the nicely humidified chamber itself.
All right. I have a question for you, actually. So given the sort of stick-like shape of your molecule, it sort of makes sense you would have a bit of preferred orientation. And I know you have some control, but not a lot of control over your ice thickness. But is there some sort of dependency between the ice thickness and what kind of views you see? Or is that something you looked into?
I want to say I'm not seeing different views because of ice thickness. We either lose contrast or we see the particles, but just the thickness of ice doesn't seem to change what we are seeing in terms of orientation that much.
Thank you. There's a question from Kai. He said a lot of the classes that were excluded from ab initio look like plausible RNA structures. Did you further investigate those via refinement?
We did some of them, but there are not enough particles corresponding to those classes to come up with any usable solutions for us. I don't know whether it's because because CryoSpark is not, these are different views that CryoSpark is not, aligning that well, or it's just particles that, I don't know how these classes, the short classes, what they are, but we did look at them, we did try to refine them, but we're not seeing anything meaningful out of them.
I have another question. So the sample you used on the chameleon, that was not the Fab. That did not include that.
Yeah, no, this was not. So the Fab bound RNA data was collected at UTMB. The sample we did at NCCAT was pure RNA, just RNA alone.
Yeah, and I guess there's always more things to try than time to try them. But do you think that would be worth trying?
So Fab was really useful, and I would love to go back to it. But another issue with the RNA samples is the complexes don't stay together when we blot them. Maybe the blot-free system is what would actually help with that. But especially when we do VitroBot, even when we mix Fab and RRE together, they don't stay together as a complex, even though Fab itself helps RRE stick to the grid or stick to the ice better.
That makes sense.
I have a question too, Keerthi.
Oh my God.
I'm very familiar with coating grids with polylysine, and that will generally put a positive charge on the whole on the carbon support. I'm not familiar with anything that would do the same thing with a negative charge. And I was thinking that if you want to chase the RNA off of the carbon and into the hole, you would make your carbon negatively charged, just like the RNA, and that would repel the RNA into the holes rather than trying to attract them to the carbon with a positive charge, that the polylysine. I just don't know of any kind of coating that produces a positive charge like polylysine makes, a negative charge like polylysine creates generally a positive charge. Do you know of any?
I don't, but when we tried detergents, that was also one of the rationales. Like it's a little hard especially when I'm coating grids, it's hard to say only stick to carbon, only stick to ice. I'm just coating the entire grid. But the idea with detergents was that somehow it would just neutralize the charge effects, whether positive or negative, and that would help push the RNA away from the carbon and into the ice. Again, all of these methods, they did work, but they were not consistently working when we tried. So that was, yeah. What did work definitely was the protein, the Fab coating.
All right. So I see one more question right now. I'll make a final call for questions. There's a couple actually. So related to charge on the grid, Preeti asked about amylamine for a positive charge on carbon.
I have not tried it. Probably. I mean, it's an amine group, so maybe. I couldn't say.
Yeah, actually, she mentioned actually she does use it a lot. So we have heard of people using it. We tend to prefer polylysine instead, just because of potential toxicity issues with the amylamine. But I think in theory they're supposed to do similar thing. All right, and Matthew has a comment more than a question. It might be a mess, but heparin is used as an RNA inhibitor because of its negative charge and similarity to RNA. So I'm not sure if that was ever on your list, Keerthi, but
there were a lot of chemicals. I
yeah.
didn't try all of them.
Yeah,
I'm pretty sure I heard of heparin too in that list.
Yeah, thanks for that tip, Matthew. All right, I think we've actually gotten through all of the questions. So thank you very much, Keerthi. That was amazing advice. All right, so thank you everyone for joining us today. Come back next month for more Cryo-EM current practices. And we wish you all luck on your Cryo-EM journeys, and the centers are here to help you. So let us know how we can help.